Okay. Okay, so hi everyone. I'm Ashley and I studied physiology at the University of Bristol. So today we're going to be talking about the regulation of muscle contraction. So just a bit about my course. I study physiology in University of Bristol, and so on the screen here are some core pieces of information you need to apply to this course in Bristol. And the entry requirements for A-levels are AAB, and for IB it's 34 points with 17 at HL. And you'll need a biology plus another science, which could be in chemistry, physics, maths, or psychology. So in terms of my experience, why I chose this course was because in sixth form, I was very interested in biology, and I was specifically interested in human biology, so not so much the plant biology or biotechnology. So I was more interested in how the body works, how different organ systems work together, and how the body reacts to certain mechanisms. So I found this course and I thought it was perfect because it really covers all of that and delves a lot into human biology. So in terms of my modules, I picked 3 optional modules and 3 compulsory modules. So that's the baseline of first year. So the 3 compulsory modules are 2 physiology modules and 1 pharmacology module. So in your pharmacology module, you learn more about how drugs react and how drugs cause your body to do certain things. It's also very interesting and quite a vital component when studying physiology as well. For my optional modules, I picked 1 biochemistry and 2 anatomy modules. So a lot of people in my course actually picked this course because it's quite like a pre-med degree. So as you can see, I picked like biochem, anatomy. So a lot of it is actually covered in medicine as well. So this course is very good in terms of not knowing what medical field you'd actually like to go into, whether if you do want to go into healthcare but like you're unsure about becoming a doctor, this would be a very good pre-med introductory course for you. So in terms of living abroad, I found living in Bristol to be a very, very good experience. If you like London but don't like the hustle and bustle of it, Bristol is definitely a very good alternative. It's more of a city university, and it's got London vibes, I guess you can say, but it's not as big, and I feel the close-knit community makes it a lot easier to make friends as well. And the University of Bristol has a program and has international student ambassadors to help you settle in. So when settling in, it can be quite daunting to get opening a bank, but these student ambassadors can greet you at the airport and help you through all those things. So I found it to be very nice and easy to settle in. In terms of accommodation, I lived in East Village. So University of Bristol has 3 villages: East Village, West Village, and North Village. I lived in the courtrooms, and the East Village is known to have more modern accommodation, and it's a lot closer to the university, so it was about a 10-minute walk. North Village is more if you are looking for a cheaper accommodation, or you're prioritizing maybe your social life as well as cost. And West Village, it's very cost savvy as well, so that is something that you could take into account of when you're choosing accommodation. It was very easy to pick and choose accommodation. You're not guaranteed your first choice, but personally I was, so I feel like it is very, very, very good and easy to find accommodation, especially if you're an international student. It's a lot easier to get the accommodation that you do want. So now I'm going to talk about the actual lecture and the regulation of muscle contraction. So when do you learn this? You learn this in your first year, and you learn this in teaching block A. So each course is divided into 2 teaching blocks, teaching block A and teaching block B. So it's basically 2 semesters. And you learn this because it's an introduction to integrative physiology. So integrative physiology is about how exercise impacts your body and how your organ systems respond to activities such as exercise, and of course muscle contraction is very important when it comes to that. So I'm going to specifically talk about calcium ions and how it's involved in skeletal muscle. So it's important to note that I'm going to be talking about skeletal muscle today because it's different, the mechanism is different in cardiac and smooth muscle. So a brief overview on what I'm going to talk about. I'm going to be mainly talking about muscle contraction. So as you can see here, there are 5 main components of muscle contraction, and I'm going to be talking a little bit about muscle relaxation as later on you can see that calcium ions play a larger role in muscle contraction rather than muscle relaxation. So you may have learned this in A-level biology already, but muscle contraction is due to something called the cross-bridge cycle. So each cross-bridge cycle accounts for one muscle twitch, which in turn counts for a muscle contraction. So the cross-bridge cycle starts off with ATP hydrolysis. So that's the breakdown of ATP. And this releases energy, so it energizes this cross-bridge over here. So once the cross-bridge is activated and energized, the myosin binding head, which you can see in blue over here, binds onto this actin filament seen in gray. So once it allows the binding, the cross-bridge actually allows the 2 filaments to move and glide past each other. And this movement is called the power stroke. So once this power stroke occurs, this is what causes the muscle twitch. So when it goes back into the relaxed state, ATP has to bind onto the myosin, which causes the cross-bridge to detach. So this step is actually the muscle relaxation itself. And how muscle contraction happens again is, again, the ATP is hydrolyzed and energizes this cross-bridge. So this cross-bridge cycle is what regulates and what causes the muscle to constantly contract. So of course, each muscle contraction starts off with something called the action potential. Again, you may have learnt this in your biology lessons. So an action potential is an electrical signal, and it's passed through, specifically for muscles, it's passed through a motor neuron via saltatory conduction. And it's passed to this end plate over here. So this end plate is just where the neuron meets this muscle fiber. So once the electrical signal is passed to this end plate, the chemical message is diffused and it causes and triggers the action potential across this muscle fiber. So as you can see from this graph, each action potential causes one muscle twitch, but there's a period between one action potential and one muscle twitch, and that's known as the latent period. So actually the contraction is not simultaneous with the action potential, and the twitch occurs after a short delay of the firing of the action potential. So this delay is too short for calcium ions to fire outside of this muscle fiber itself, and it causes the contraction to occur after a delay. So this is not vital for the body because the contraction won't happen directly after this message has been sent. So how does the body accommodate for this? So we have a calcium storage actually inside our muscles fibers itself, and it's stored in something called the sarcoplasmic reticulum. So the sarcoplasmic reticulum is this web-like structure over here, and it surrounds myofibrils. So myofibrils are small cylindrical structures in each muscle fiber. And each sarcoplasmic reticulum is surrounded by this yellow structure over here, and this yellow structure is a T-tubule. So I'm going to go a bit into the sarcoplasmic reticulum. So the sarcoplasmic reticulum, as I mentioned before, stores these calcium ions, and it stores the ions specifically in a protein called calsequestrin. So the calcium ions in your cytosol or in your cells get moved into the sarcoplasmic reticulum. And it does so by this protein called calcium ATPase, and via the energy breakdown from ATP hydrolysis again, it allows the movement of these calcium ions to move into calsequestrin, as you can see in this animation. So once it's stored in calsequestrin, how is it moved back into the cytosol for muscle contractions? So it's moved out via this receptor called the ryanodine receptor, and this receptor allows the movement of calcium ions out. as you can see here. So once these calcium ions come out of the sarcoplasmic reticulum, it gets ready for the muscle contraction. So as I mentioned before, there are these structures called T-tubules that separate each sarcoplasmic reticulum. So the function of these T-tubules is to conduct the action potential from the sarcolemma, so from the surface of the muscle fiber deep down into the sarcoplasmic reticulum. So the muscle fiber is about 50 to 150 micrometers in diameter. So the action potential isn't able to reach deep inside the sarcoplasmic reticulum and have access to these calcium ions. So these T-tubules help the action potential go in deeper into this muscle fiber. So as you can see here in this diagram, this dumbbell shape is the sarcoplasmic reticulum, and this over here is the T-tubule. So of course it starts off with this action potential. So the action potential, because of this T-tubule, can go deep inside. And once it goes deep inside and it has close contact with the sarcoplasmic reticulum, it allows the calcium ions to be able to activate this muscle contraction. So when it reaches the sarcoplasmic reticulum, it actually causes a receptor that is present on the T-tubule called the dihydropyridine receptor. And this receptor is like a voltage sensor. So when the action potential reaches deep inside, it causes this receptor to change conformation. And because this receptor is coupled with the ryanodine receptor, the change in conformation allows the ryanodine receptor to open. And because this ryanodine receptor opens, it then allows the calcium ions to flow out and flow into the cell and out of the sarcoplasmic reticulum. So, as I said before, muscle cells are made of many little myofibrils, and each myofibril has thick and thin filaments, as I said, which are the actin and the myosin. So as you can see here in this diagram, the actin are the small green spheres, and for the cross-bridge cycle to occur, the myosin has to bind onto this actin. However, there are two components that are actually blocking this actin from binding with the myosin. So the myosin binds onto the actin binding site, the myosin binding site on the actin, but two complexes are bound onto this site, which prevents the binding of myosin, which then leads to the prevention of the cross-bridge cycle. So what are these two structures? So the first structure is known as tropomyosin, and for simplicity, I'm going to call this the rope structure. So this rope structure is bound onto the binding site. And also this troponin complex, which is the second structure, is also bound onto this binding site. So this troponin complex is made up of three components. First is the TNT, which is bound onto this rope structure, the TNC, which allows the binding of calcium, and the TNI, which is the inhibitory complex. So the inhibitory complex and this rope structure is the ones that are specifically bound onto this binding site, which prevents myosin from binding. So this is a zoomed in structure and a cross-section of what I showed previously. So again, these two spheres are the actin. And the TNI, the inhibitory complex, is bound onto this binding site as well as this rope structure over here. So how does this whole structure, including this rope structure, move out of the way in order for myosin to bind? So two calcium ions are bind onto TNC because it allows the binding of calcium. So once two calcium ions bind onto TNC, this whole structure is able to move. So this rope structure over here binds onto this groove, as you can see over here. And once it binds onto this groove, this whole structure is able to move out of the way, as you can see over here, and therefore exposes this white bit, which is the binding site, which then again allows the myosin to bind onto the site, which starts the cross-bridge cycle, which in turn allows muscle contraction to occur. So that is the main role of calcium ions in muscle contraction. So in terms of relaxation of the muscle, the excess calcium in the cytosol is then pumped back into sarcoplasmic reticulum and is stored in the calsequestrin until another action potential arises. and until your muscles need to contract again. So the excess calcium that is moved back actually restores the inhibition. So it's no longer bound onto troponin, and it allows the whole complex to be bound onto the site, which prevents the cross-bridge cycle from happening. So to make this a bit simpler, I'm going to describe a little situation. So what happens when you're dunking an Oreo into a glass of milk? So the muscles that contract in this scenario are actually the muscles in your fingers when holding the Oreo, as you can see over here. So when your fingers hold the Oreo, the muscles have to contract to be able to hold the Oreo in place. So I'm going to show you this little video. So as you can see here, this blue thing is actually the action potential, and the muscle fibers are stimulated by this nerve impulse. And when it's stimulated, it again causes the increase and the exit of calcium ions from the sarcoplasmic reticulum. So there's an influx of calcium ions, which are seen over here in yellow. And these calcium ions bind onto the troponin complex, specifically TNC. And once it binds, it allows the movement of the rope structure and troponin, which then exposes these binding sites seen in dark purple. So once these binding sites are removed, now the myosin can actually attach onto these binding sites. So it causes the release of ADP and phosphate to allow the binding of these two, as mentioned previously when I talked about the cross-bridge cycle. So because of this binding, it allows both filaments to glide past each other, which causes the actual twitch itself. And then in turn, an ATP molecule then binds onto this myosin, which causes the muscle to relax. And it causes the myosin head to stop binding onto this binding site. So you can see here that ATP binds onto the myosin, which then causes the release of myosin. And then the ATP is broken down again into ADP and phosphate, which then goes back into its normal relaxed state. So I've included a little summary here on the whole mechanism of muscle contraction. So these lecture slides are going to be sent to you, so you can use this for notes and just as a quick brief summary of what happens and how calcium ions actually help in muscle contraction. So in physiology, it's also very important to talk about clinical applications as well as situational applications. So in this case for muscle contraction, a clinical application and a disease that can arise from this is Brody disease. So Brody disease is caused from a prolonged calcium ion elevation, and it does so because there's a mutation in a gene, and this gene allows the transportation of calcium ions from the cell into the sarcoplasmic reticulum. So because this gene is mutated, it can't work. So there's going to be an increase in calcium ions in your cell because it can't move into the sarcoplasmic reticulum. And because it's always in your cell and there's a constant rise of calcium in your cells, it will constantly be bound onto troponin. And because it's bound onto troponin, the myosin head will always be attached onto the actin, which causes the muscle to contract constantly. So as you can see over here, this man, when he puts his hands in a bucket of ice, his hands are constantly contracted. And as you can see here, when he's walking, he's very stiff. And that's because his muscles are always contracted, and it's because of the elevated calcium ions. And because of this, it also leads to the slowing of relaxation. So when he walks, it's going to be very stiff. So yeah, that's the end of my lecture series. If you have any questions, you can contact me via this email here. Or if you have any general questions, you can contact the Southeast Asian office over here as well. Or you can ask me any questions now.
Thanks so much, Ashley. That was really insightful and interesting to know how much work goes into dunking an Oreo into a glass of milk. So we have some questions. Feel free to post more participants if you have any. One student has asked, what kind of jobs do physiologists do in the medical profession?
So there are a lot of jobs in the medical profession that physiologists can do. So um, usually, well, most of the time, um, you can either go into graduate medicine, but if you don't want to become a doctor, there's something called a physician's associate. So this is a 2-year master's program that actually a lot of people from physiology go into. And a physician associate is like a doctor, but they work a lot more closely with the nurses and other professionals in the healthcare system. Also, physiologists can go into research because research is a very important component, especially in the medical field. Physiologists have a very good insight because we delve right into how the body works, and we go into a lot more detail than, let's say, biomed, for example, because we're just learning about the organs and about the systems in general, rather than cells and how like photosynthesis works, for example. So there is a lot of careers like that you can go into as well.
Thank you. And what do you think you might be doing, Ashley, after you complete your degree? Have you thought much about that?
Currently, I'm not too sure, but I will definitely continue with physiology in the future and something science-based.
Great. Another question is, could a physiologist work in the nutrition sector? Could you work?
Yes, of course.
Yeah.
Right. Yes, you can work in the nutrition sector. So, because nutrition is a lot about how bodily organs work and how bodily systems work in relation to food and carbohydrates, amino acids, etc. You can definitely go into that. So there's also a nutritional course in Bristol for masters, I think. You may have to check on that, but you can go into nutrition as well. Actually, you can go into a lot of different things, but I feel like learning physiology would be a very good base and a very good introduction to nutrition in the future.
Lots of questions about jobs. So what do you think is the job market for students in life sciences if I was thinking of doing biomedical science or physiology or something else in the field?
So, in terms of the job scope, I feel like you can really do a lot of things. So, not only is there healthcare related things, but there's also research. And there's a few students in life sciences faculty that actually go on to do other things, such as finance or even law, even. So, there's a lot of things. And because science is so broad, you can really do anything. Because you're graduating from a BSc, and I understand that a lot of companies actually look for people with BScs at the moment, so there's a lot of different things that you could go into.
It's good news to hear, especially when the economy is in this way.
Yes.
So I am not taking chemistry at A levels. Will that disadvantage me when I start doing the life science course?
Okay, so I actually didn't do chemistry either, and physiology doesn't actually need chemistry as an entry requirement. It is advised to take chemistry A level, but personally, as a person that didn't take chemistry A level, I found the content to be okay, as in it was quite all right to grasp. It was initially difficult, especially the pharmacology side, because that was where most of the chemistry comes into. And because I also chose biochemistry as my optional module, that was quite tough in the beginning, but you do get a lot of help, especially in more chemistry based topics. You actually have labs, more labs and more tutorials where you can ask your lecturers and professors for a lot of help. So I wouldn't think it's a downfall if you didn't take chemistry level.
Thanks, Ashley. So I think the questions have stopped coming in. Maybe we'll give it a minute more if no one else has any questions, but let's have some parting words from Ashley. Do you have any advice for prospective students who are about to embark on a life science degree and maybe also come to study at Bristol?
So Bristol's definitely amazing. It's honestly been one of the best years of my life studying in Bristol. It's honestly been the best. It can be very daunting at first. I was very scared, and of course, because of COVID, it must be a lot scarier to apply to countries abroad. But I assure you that Bristol is very, very nice, and the university has actually taken a lot of precautions in terms of COVID. So I wouldn't be too worried about that. In terms of life sciences, I think my tip is to really focus on biology and perhaps chemistry as well in your A levels, and find what you really truly like, because in life sciences there are so many different courses that you can go into, such as pharmacology and neuroscience, which are a lot more niche as well. But if you like a more general sciencey biology based course, biomed is also another good option. It's not as niche as physiology and pharmacology, but it's another good option to go into.